Dear students and my teacher friends, Namaskar. I welcome you to the presentation lecture on digestion of fat. Now, as you can see on the screen, I am showing you a few chemical formula or the structure of the dietary fats, the lipids which are present in our food. So, this is the information, chemical formulas of some common lipids. Now, here on the left hand side, this is the fatty acid, and we know in the structure of fatty acid, this one is the COOH carboxylic acid group, and this one is the hydrocarbon chain, and between the carbon single bond is present, then it is the saturated fatty acid. And if in the structure of fatty acid, if double bond is present between the carbon, then that type of fatty acid is known as the unsaturated fatty acid. Now, this is the structure of glycerol, and you see in the structure of glycerol there are 1, 2 and 3 OH, that is the hydroxyl groups are present, and that's why this glycerol is known as trihydroxy alcohol. And the symbol, this is the symbol for the glycerol. Then tri-acyl glycerol. Now, if you observe this structure, you will find that in the structure of tri-acyl glycerol, there is one glycerol is there, and with this, 3 fatty acids are attached to it. So between the 3 fatty acid and the glycerol, the bond which is present, this bond is the ester bond. Then from the structure of triacyl glycerol, if we remove, let's say if we are removing one fatty acid, then the structure will be known, instead of triacyl glycerol, now since we have removed one fatty acid, the structure will be now known as diacyl glycerol. Then if we further remove one more fatty acid, that means only one fatty acid is attached to the glycerol molecule, so now we are having monoacyl glycerol. Then phosphatidyl choline, lysophosphatidyl choline, cholesterol, cholesterol ester, cholic acid. So these are the main dietary fats or the common lipids of our food. So now we are starting this lecture. This is the first point. Digestion of fat is the title. Dietary fats. Types of fats. Fats are of 3 types: simple fat, neutral fat. Example of simple fat, triglyceride and the cholesterol. Compound fat, phospholipid, associated fats, steroids and fat soluble vitamins. Now the dietary fat, the food that we eat, the lipid which is present, Okay, so the dietary fat is from the vegetarian food. We know some people are vegetarian and some people are non-vegetarian. So vegetarian food, let's say we are getting the lipid from the butter or from the oil or from the ghee. Okay, and from animal origin, let's say the meat and the eggs. Okay, in that the dietary fat is present. Now the fat that we consume, it is in the form of neutral fat. That is, the fat is in the form of triglyceride mostly. Then in the dietary fat, phospholipids are present, cholesterol are present, some free fatty acids are present, lecithin and cholesterol esters are also present. Now, daily intake of fat, how much how how much fat that we consume in a day. So 25 to 160 gram of fat we are taking daily. Now moving on the second point, digestion digestion of fat, site of digestion, where exactly in our body the process of digestion of lipid dietary fat is going on. Now in the mouth, secretion of lingual lipase take place, and in the stomach secretion of gastric lipase takes place. Now both this enzyme, lingual lipase and gastric lipase, they are not significant, and practically we can say they are not playing a role in the process of digestion of dietary fat. Or in other words, we can say that in the mouth or in the stomach there is no dietary fat digestion process is taking place. So that is the meaning. But few traces of dietary fat, few traces, they are digested in the stomach. Okay. So now the question arises, so where the process of digestion of dietary fat is taking place? So now we are discussing the third point now, the role of stomach, the role of stomach in the digestion of dietary fat. Now stomach empties the chyme. Now we know what is chyme, the food which is digested in the stomach, the acidic food, that is known as the chyme. And the stomach is emptying this chyme slowly into the small intestine. So we can say in the small intestine the chyme remains for a longer time. So as the chyme is remaining in the small intestine for a longer period of time, what happens? The pancreatic juice, the enzymes of the pancreatic juice, the lipases, they can act on this chyme. That means the fat which is present in the chyme. So the enzyme, they can act on this chyme. So slowly, the process of lipid digestion is taking place in the small intestine. So the role of the stomach is to empty the chyme slowly into small intestine. Now, how this chyme, what is the factor? Why this chyme is entering slowly into the small intestine? So, this process is slowed by CCK hormone. And when this CCK is secreted, so the dietary lipid when first appear, they first enter into the small intestine, that time the CCK is secreted. And this CCK is slowing the process of emptying the chyme into the small intestine. So the small intestine will get time to digest the dietary fat. Now moving on the fourth point of our discussion, process of digestion or the mechanism of digestion. Now in the process of digestion of fat, there are three steps. First step is the emulsification of fat by the bile salt. Second step is the hydrolysis of fat by pancreatic and intestinal lipolytic enzymes. And the third step is the micelle formation. So we are discussing one by one each step. This is the fifth point that we are discussing now. Emulsification of fat by bile salt. Now, what is the meaning of emulsification? So, breaking of large fat drop into the smaller droplet is the prerequisite for the action of pancreatic lipase. Now, what is the meaning of this point? Now, this is the fat globule is shown, and as you can see, this fat globule is large in size. So since this fat globule is very large, the pancreatic lipase cannot act on this larger fat globule. So the dietary fat that we ingest, that is in the form of large fat globule, and on this fat globule the pancreatic lipase cannot act. Now, pancreatic lipase is the water soluble enzyme, okay, and since this pancreatic lipase is water soluble, it is difficult for this enzyme to act on this larger fat globule. So how this will act on this fat globule, that is the question. So the action of pancreatic lipase prefers oil water interface of fat, and with this interface the surface area increases for the action of lipase, and this surface area increases 1000 times, and the mechanism for increasing the surface area is known as the emulsification of fats. Now what happens in emulsification of fat? Now, which is the factor responsible for the emulsification of fat? So the bile salt are responsible for the emulsification of fat. Now, what these bile salts are doing? So they are lowering the surface tension of this. So here bile salt, and they are decreasing, they are lowering this bile salt, they are decreasing, they are lowering the surface tension of larger fat globule. And this action of the bile salt is detergent like action. Now what is happening over here, that the surface tension of the fat globule has decreased, okay, and at the same time in the small intestine the segmentation movement is also taking place. So now there are two processes. Bile salt, they are reducing the surface tension, and the small intestine, the movement of small intestine, the segmentation movement. And due to this movement, this fat globule is broken down, and this fat globule breaks up into the smaller droplet, and these droplets are having the size of 1 mu m, 1 micron. Okay, now here you see the figure. So by observing the figure you can understand the mechanism, what is happening in the emulsification. So the large fat globule is shown, and here with a green color bile salt is shown, and the phospholipid is shown. Now you see the bile salts and the phospholipid, they attach to the surface of the fat globule, and they are converting this larger fat globule into the small lipid droplets. And this small lipid droplet, on their surface, as you can see, the bile salts are present and the phospholipid is also attached to this smaller droplet, and this process is known as emulsification. Okay, now what is the usefulness of this process, emulsification? So this is the droplet. Okay, so now there are so many small lipid droplets, and since these are small, you can see the surface area has increased now, and now the enzyme pancreatic lipase, now this enzyme can act on this smaller fat droplets. Now one more thing, component of bile, lecithin. Now what the role the lecithin is playing? So lecithin is performing the stabilization action on the emulsion, and it is also enhancing the emulsifying action of bile salt. So what is the meaning of this thing? That these are all small fat droplets. Okay, and it is possible that again this small droplet, they join together and they can form, once again they can be converted into larger fat globule. But the lecithin, a component of bile, is preventing this thing. So this small droplet cannot merge together once again, and they cannot form the larger fat globule. So this is the 6th point. Now moving on the 7th point. Now before that, I would like to show you this entire figure. So process of emulsification. then this, okay, information. And here bile salt, the structure of bile salt, glycolic acid, and here this one, structure of bile salt, this side the polar side, and this side is the non-polar side. So this side hydrophilic side, this side hydrophobic side. Non-polar side, that means having attraction for water, okay, and the sorry, here I have, by mistake I have made the mistake, I apologize for that. This is the polar side, okay. So this is the hydrophilic one, and this side is having, this one is having attraction for the water, and this one is the non-polar side. So this side is the polar side, is the hydrophilic, and this one is the hydrophobic, okay. Polar side hydrophilic, hydrophobic. So this is the information, okay. And this is the structure of bile salt. Moving on the 7th point of our discussion. Now the attachment of bile salt, how they are present around the small fat droplet. Now, as I was explaining you, that the bile salt, they surround the fine fat droplet in that manner that their lipophilic non-polar end are toward the fat, okay, and the hydrophilic polar end, they are towards the aqueous phase. Now what is the meaning of this thing? So once again, here you see polar side, and you observe closely, this is the polar side, okay, and where this polar side is attached, okay, so this polar side is here, so it is present outside, and this is the non-polar side, this is the non-polar side, so this is the non-polar side, this one is the non-polar side, so this is the hydrophobic side, and this non-polar side is attached to the fat, and the polar side, this one is the polar side, so this polar side is toward the aqueous phase. Now here we have to understand that over here the pancreatic juice is there, and in the pancreatic juice there is a water, so this side, which is the polar one, is towards the pancreatic juice, that is the aquatic phase. So this is the 7th point. Moving on the 8th point. Now the 8th point. Now in 8th point, now we are discussing the second phase of the digestion of fat, hydrolysis. Hydrolysis of fat droplet by the pancreatic and intestinal lipolytic enzymes. Now the pancreatic lipolytic enzyme. Pancreatic juice contain three types of lipolytic enzymes, and this lipolytic enzyme, they hydrolyze the fat. Okay, now let's observe this figure and let's try to figure out the emulsification process. So this is the title: emulsification. Large fat globule. Over here, this one is the bile acid and this one is the lecithin phospholipid. So this bile salt and lecithin, they break this larger fat globule into smaller fat droplet, and the surface is covered by the bile salt or the bile acids. So here in the bile acid, you see the blue color portion, the blue color region, this part, this one is the hydrophilic region, and the white color portion, this one is the hydrophobic region. So if you observe closely, you will find this is the hydrophobic, and hydrophobic region of bile salt or the bile acid is in a contact with the small fat droplet, and this blue color region, this one is the hydrophilic region, and this region is towards the aquatic phase, towards the pancreatic juice. So fat globule is broken up and coated by lecithin and bile acids. Now moving on the ninth point of our discussion. This is the ninth point of our discussion: pancreatic lipase enzyme, the action of pancreatic lipase. How pancreatic lipase digests dietary fat. Now, pancreatic lipase is a powerful, very powerful lipolytic enzyme. And colipase, what is colipase? Colipase is a protein which is present in the pancreatic juice. And what is the function, what is the role this colipase is playing in fat digestion? So this colipase displaces the bile salt from the smaller fat droplet. So here you see the small droplet on the surface, the bile salt are present, and the colipase, the action of colipase, it is removing this bile salt. So once the bile salt are removed, pancreatic lipase enzyme can act on the small fat droplet. Now the pancreatic lipase hydrolyzing almost all the triglyceride, the neutral fat of the food. And when this lipase, pancreatic, when it hydrolyzes the fat, it produces two fatty acids and a monoglyceride. So with the figure, we are trying to understand this point. This is the ninth point. Okay, in the figure, you see this point, fat hydrolysis. Now colipase protein, colipase first displaces the bile acid. So now these bile acids are removed. So small fat droplet, this one is open now. So now pancreatic lipase enzyme, which is shown with the red color small triangle. So this enzyme now acting on the dietary fat. And how this enzyme is acting? So the action of pancreatic lipase is shown over here. This is the structure of triglyceride. Structure is shown, and we know in the structure of triglyceride, this is the glycerol, and this one is the 1, 2 and 3. These are the 3 fatty acids. Now what this pancreatic lipase is doing, so this pancreatic lipase is breaking the ester bond, this one and this one. Okay, so since the pancreatic lipase is breaking the ester bond, what happened? This fatty acid is released. So fatty acid number 1 and this fatty acid is released. Okay, fatty acid number 2 and fatty acid number 3, the third one is released, and the second fatty acid remain its place and it is attached with the glycerol. So this attached molecule, the glycerol attached to fatty acid, this molecule is known as the monoglyceride, and now we are having 2 free fatty acids. So see over here it is written emulsification droplet, they are acted upon by pancreatic lipase. Pancreatic lipase hydrolyzes first and third fatty acid from the triglyceride, and it is not removing the middle fatty acid, the pancreatic lipase. So this is the ninth point of our discussion, and for the ninth point, this is the figure. Okay. Now moving on the 10th point. Now in the 10th point, enzyme cholesterol ester hydrolase. Now the cholesterol which is present in our food, the dietary cholesterol, is in the form of cholesterol esters, and this cholesterol esters are hydrolyzed to cholesterol and fatty acid by the action of enzyme cholesterol ester hydrolase. So here in the figure, digestion of lipid, cholesterol ester, enzyme cholesterol ester hydrolase. This enzyme acting upon cholesterol ester, and it is hydrolyzing cholesterol ester into cholesterol and fatty acid. Now, the cholesterol ester hydrolase is secreted as an active enzyme, and it is hydrolyzing the cholesterol ester to free cholesterol and the fatty acid. And this enzyme also hydrolyze the ester bond, the ester linkage of triglyceride, and this can remove all three fatty acid, and then this can provide the glycerol. So triglyceride can be acted upon by this cholesterol ester hydrolase. All three fatty acid can be removed from the triglyceride, and we can have the molecule of glycerol by the action of cholesterol ester hydrolase enzyme on the fat. Then point number 11, phospholipase A2. Now this phospholipase A2 is secreted in inactive form, and then it is converted into active form. Phospholipase A2 secreted as a proenzyme, and like many other pancreatic enzyme, it is activated by trypsin. Now, what is the action of this phospholipase A2? So here, this one is the phospholipase. Phospholipase A2 hydrolyze this phospholipid, and it converts this phospholipid into lysolecithin and fatty acid. Now, as we have learned the entire mechanism or the entire process of fat digestion, so the product of lipid digestion are monoglyceride. Monoglycerides are formed after fat digestion. Fatty acids are formed, cholesterol is there, lysolecithin, glycerol is also there. And the glycerol, right now we have we have studied that it is formed by breaking the ester bond of triglyceride and by releasing the fatty acid. Now the glycerol, with the exception of glycerol, each end product is hydrophobic and therefore is not soluble in water. The only exception is the glycerol. The rest of the product is hydrophobic and they are not soluble in water. So now it is important that this hydrophobic digestive product, all this product, they must be solubilized, and this product, all this lipid digestion product, they are solubilized in the form of micelle. And once the micelles are formed, these micelles, they are transported to the apical membrane of the intestinal cells for the absorption process. So this is the third and final point of our discussion, that was the micelle formation. Then the last point. Now here in the last point, 12th point, intestinal lipolytic enzyme. Now the brush border of epithelial cell, they contain small amount of lipase and cholesterol esterase. So I am drawing, let's say this is the epithelial cell of small intestine, the enterocyte, and this is the apical border. And on the apical border, the microvilli are present, and together all the microvilli, they appear like the bristle of brush. So that's why this border is known as the brush border. And on this brush border, enzymes are present, and this enzyme, the name of two of them, that is the intestinal lipolytic enzyme, okay, lipase is present, and cholesterol esterase are also present. So the effect of this two brush border enzyme is same to the pancreatic lipase. The manner in which pancreatic lipase act on fat triglyceride, that it is breaking, it is removing the first and third fatty acid, the same way this enzyme are also acting on the fat. So this was the last point of our discussion for this topic. With this, we have completed the discussion on digestion of lipid, that is the dietary fat. I hope you have enjoyed this presentation lecture of lipid digestion, and I also hope that this presentation lecture will be helpful in your exam preparation and also in your studies. My name is Manish Kosti sir. I am from Ahmedabad, India. Bye bye. Namaste.